last points we took was promoter, operator, turn on, turn off mechanism. Beside this, prokaryote gene regulation. Prokaryote gene regulation, we did cell specialization and transcription. The 2 points we did. Now we are going to start from the RNA interference. Right? The new topic which we are going to take today is about what? It's about RNA interference. Have you ever heard before about this RNA interference? Okay, small circular RNA. We studied it. Why not? We studied it. We don't have it in the book, but we have studied. When we were studying the type of RNA, right? So at that time, I remember I told you we have 3 major type of RNA. What are they? 3 major type of RNA. RNA, right? 3 RNA we have: ribosomal, transfer, and messenger. Besides this, we also studied another type of RNA, but I told you at that time that this is not in your book, right? On this level, we are not going to study that type of RNA, which is sRNA and which is RNAi, right? sRNA is small RNA present in the cell, right? Besides this, we have some small other, we can say, fragments of the RNA, which is called RNAi or interference RNA interference, right? The small sRNA we are not studying, but interference RNA, it's important, so we will take it, right? For ear biologists wondered why cell deaths contain lots of small RNA molecules, only a few dozen bases long and don't belong to any of the major group of RNA, which are messenger RNA, transfer RNA, and ribosomal RNA. If you look in the cell on the molecular level, right, under the microscope, inside the cell and inside the nucleus, in the cell we have many small RNAs, right? We are saying that in one sense, the fragments of RNA, pieces of RNA, right? The pieces of RNA which are round about little bit, like a few dozen, like 24, right? 2 dozen, 3 dozen base pairs, right? Or 4, 5. That is not too much, right? It is just a fragment of RNA, and it's not 1, 2, 3 in the cell. It's a lot in number in the cell, right? But actually, this RNA, it is not going to belong to any of the known 3 groups. It is neither tRNA, it's neither mRNA, it is even not ribosomal RNA, right? So 3 major types we have, but these small interference RNAs or the fragments of RNAs, they are not going to be counted in any type of the RNAs. These small RNA molecules play a powerful role in the regulating gene expression by interfering with the messenger RNA. Now, it is not going to be the part of any type of RNA, but it has an important function, right? It has function. And what is the function? The function it has that when these RNAis, they are going to interfere, they are going to interact with the messenger RNA. So they are regulating or changing the gene expression, right? They are regulating and they are going to change the gene expression. And it's also believed that it's going to initiate this gene expression, right? These small fragments. After they are produced by the transcription, the small interfering RNA molecule fold into a double-stranded hairpin loops, right? If you look in the diagram, right? So when it is being formed, after the formation of these interfering RNA, when it is being formed, after that, as we know, it's a short RNA, right? It's made from a few dozen base pairs. So when they are formed after their formation, they form what? They form a double. RNA is always a single strand, right? RNA is always single strand. They form a loop-like structure, right? And it's called a hairpin loop. You can see this is one strand, right? Complete we have one strand, right? This, but it form a double stranded like structure. So this place where it form a double stranded like structure, it is here pin loops, right? Or we can say loop like structure. It form what? It form loop like structure. An enzyme called dicer. Enzyme cut or dice these double-stranded loops into microRNA. Right? Each about 20 bases pairs in the length. The two stranded of the loops then separate. Now, when it formed the hairpin loop-like structure, right? When it formed the hairpin loop-like structure, so after that, there is an enzyme, right? RNA polymerase, right? So in RNA polymerase, to be specific, it we have dicer, right? But those enzymes which are acting on the RNA, that's called our RNA polymerase. To be specific, we have enzyme dicer. Now the function of dicer is what? The dicer is going to cut the hairpin loop-like structure into two parts. Mean the interference RNA which we have. Let's suppose this is one strand, right? This is a complete, we have to check, this is a complete long strand, but it will make a hairpin loop-like structure. And now it is looking like the DNA, right? It isn't looking like the DNA. Now it is not the DNA, it's just a short making of 3-4 dozen of base pairs. So now what will come? The dicer enzyme which are RNA polymerase, it will occur and it will cut this RNA, which we have RNA interference into two parts, right? And it will be round about how much long? 20 bases long, right? Now each of it will contain what? This is, let's suppose, one. So each of it will contain now how much? Micro RNA, micro means small, right? Small RNA which will around about have how much? Base pairs, it will round about has 20 base pairs. You can see like this, right? Let's suppose you have one, which is being cut with the help of the dicer enzyme, and dicer enzyme give us two small micro RNAs. Splicing complex, we have a head that you will understand later. Okay, one of the microRNA pieces attached to a cluster of protein to form what is known as splicing complex, right? Slicing or splicing, the previous lesson we took, right? Splicing or slicing we took in the previous lesson. The splicing complex bind to and destroy many messenger RNA containing sequence that is complementary to the microRNA. So now if you look here, right, one of the microRNA pieces attached to a cluster of protein. So when the piece of microRNA, right, microRNA mean that this RNA, right, which the interference RNA, which is cut by the dice enzyme into two parts, each is called microRNA. Now this microRNA is going to attach to a protein, right? It is going to attach into the cluster of protein. So when it attached to a cluster of protein, we called it what? We called it splicing complex. What? Slicing is what? Slice means to cut something, right? Slice or splice to cut something. So splicing complex, it is going to cut something. What it's going to cut? It is going to cut the RNA, the bases, right? The splicing complex bind to and destroy any messenger RNA. So it is going to cut messenger RNA containing a sequence that is complementary to microRNA. Which RNA will be cut? Which messenger RNA will be cut? That messenger RNA which have same basis, opposite basis to what? Opposite basis to the microRNA. Like let's suppose this we have AUG. Let's suppose in this we have what? In this we have AUG. So it will cut the messenger RNA which contain opposite of A is
UAC.
Huh?
UAC. Because the rest of not that timing is RNA and not DNA.
Yes. So it will be
UAC.
UAC, right? And messenger RNA, because in the RNA there will be no timing, right? So it will be UAC. So where in the messenger RNA UAC occurs, so it will cut at that point. Let's suppose GCC, right? So for GCC it will be CGG, right? Opposite to that. Complementary mean opposite, which is going to combine. The microRNAs stick to the certain messenger RNA molecules and stop them from passing on to the protein making instruction. Now we are saying that if you look to the microRNA or RNA interference RNA. So now if you look to the function of interference RNA at this point, right? So it is working as what? It is working as switch off, right? When it's going to bind to the messenger RNA molecule. So sometime it is going to stop them from passing on to protein making instruction. So protein making instruction is what? It is the last function of the gene expression, right? When gene will express, it will convert into protein, it will form protein. And this microRNA, when it's going to combine with a certain messenger RNA, so it is going to switch off it. So switch off, it means that it do not have function anymore. So meaning the example, if you take the first example which we took on E. coli, right? So let's suppose an E. coli is going to enter to the environment where there are no lactogen, right? Or lactose is not there. So then the microRNA is going to bind to the messenger RNA there, and it will just stop it from translation into the protein, right? You remember we studied this, right? That if E. coli enter to any environment where there is no lactose or lactogen, so what will happen? Then the gene which is responsible, the messenger RNA will not be able to transcribe and the gene will switch off. So how the gene is going to switch off? With the help of microRNA. Blocking gene expression by means of microRNA splicing complex is known as RNA interference, right? So it is called what? It is called RNA interference. The entire process is shown here. You can see, right? What will happen in the first point? In the first point, we have what? We have RNA interference or RNAi, right? RNA small I. RNA R will this will be capital and instead if you write here small I, so we call it RNAi. Right? So what will happen? It will be made from how many base pairs? Around about few dozen, right? Like maybe 2, maybe 4, maybe 5. Right? But we will take round about 4, right? 4 doesn't mean 48. 48, right? So why we are going to take it 4 roundabout, right? It's not sure that we will take compulsory 4. If you look here, when the dicer enzyme comes, right? The dicer enzyme comes. So the function of dicer enzyme to cut it into 20 base pairs, right? So it at least one part should be 20, and other if it's a little bit more than 20, so it's also okay. So it will be round about 4 dozens base pairs, or it can even exceed or decrease. But 20, when it's cut into 20, and this portion is called what? Hairpin loop, right? When the interference RNA it's made a double strand and it's formed a loop-like structure. This loop-like structure is called what? Hairpin loop. Hairpin loop. So when hairpin loop is formed, after hairpin loop, the dicer enzyme will come and the dicer enzyme will cut that mRNAi into pieces. And it will form what? It will form microRNA. You can see, right? This is cut, it gets microRNA. Now, after microRNA, what will happen? After microRNA, a splicing complex will form. And splicing complex is actually what? It is microRNA plus a cluster of protein, right? So when a specific cluster of protein combined with the microRNA, it will form what? It will form a splicing complex. And splicing complex is what? Splicing complex is made from sequence, right? From bases, right? And when this splicing complex, suppose I give you example, it will target the messenger RNA, that messenger RNA, which have complementary bases to this splicing complex, or the microRNA of the splicing complex. So when it's going to see any messenger RNA which has a complementary basis to this splicing complex RNA, microRNA, so it is going to combine with the it's going to combine with what? It's going to combine with that messenger RNA and it will turn it off, right? So when that microRNA combines with the messenger RNA, so it will stop it from working. So we are saying that the splicing complex is a destroyer. It is a destroyer. It's going to switch the genes off or it prevent the transcription or protein synthesis. Clear? At first, RNA interference seemed to be a rare event found only in a few plants, and other species. It is now clear that RNA interference is found throughout the living world, and that it is even play a role in the human growth and development. Till now, not till now. Before when the microRNA, nucleon RNA and RNA interference, when they were just discovered at the start. So it was believed that these RNA are not of important functions. And the first function was discovered that it is going to switch on and switch off the gene. But now it has been identified that it has a lot of function, right? Related to like human growth and development. The developments which are in human, it has been controlled. Mean if it is helping in the gene expression, right? So mean it almost is play a very important role with the protein synthesis. And protein synthesis is what? It is growth, it is development, and the function of our body are carrying that all are being controlled by this. So mean it is going to switch off and switch on. Now, if you look to this switch off and switch on, right? Now, what the scientists are trying to find that interference RNA which is responsible for the cell uncontrolled growth, like in the case of cancer. So, if it is found, right, if scientists identify it, so then the treatment of cancer can come out. That which interference RNA is responsible for it, right? So, as like now we are in the case of COVID, right? What we are doing, we are extracting vaccines, right? So vaccine, then that interference RNA, which is responsible, if you inject it to a cancer patient, so then he or she can recover from it. But if you find it, right? So then it can be.
Vaccine necessary the solution the RNA.
I'm talking about interference RNA. Like vaccine, I give you example. What we are doing with the help of vaccine, we are activating our body, right? But here, if we take this, right, interference, the function of interference is what? To stop gene, right? To turn off the gene. And what happened in the cancer cell? Development, continuously development occur. So when you find that interference RNA, which is responsible for that rapid growth of the cell, when you inject that interference RNA to a cancer cell patient, so it can be treated. Then the cell will not grow automatically. The cell growth will stop because the function of RNA interference RNA is to switch off the gene. So if it's fine, so it will be then.
Where we can find it.
It is difficult. It's so difficult because it's a very small fragment, and in the cell we have many small interference RNA, and it varies from cell to cell. We are not talking inside a human or animal body. We are studying it inside the nucleus, so it's very small. But still, for the best, as I told you before, in which class I told, when HIV is detected, the first case, how much time before? Too much time before, right? But round about 3 or 4 months before its vaccine has been prepared. 3-4 months before. HIV. Now it is ready for trial. Now, just I think 3-4 days before, I saw a research on this that vaccine, which is meant for HIV, is ready for the trial. So it's take years. It's take a lot of time. In case of the COVID, most of the students are asking why the COVID vaccine are prepared quickly, right? So it is pandemic, right? It was affecting the entire world. So that's why the world scientists, they sit to get the solution, to find the solution, right? But if we are talking about cancer, if we are talking about HIV, so if we have 100 scientists, out of 100, 1 or 2 are working on these topics, right? If all of them work together, so it can get good results in less time. But every scientist has its own interest, his own interest. Okay, now we have the promise of RNA interference technology, right? RNA interference technology. The discovery of RNAi has made it possible for the researcher to switch genes on and off It works simply by inserting double-stranded RNA into a cell, right? So now it's make it possible for the scientists to turn on the gene and turn off the gene, right? So on base of this, I told you that this technology, which belongs to RNA technology, or we are saying gene technology, right? So based on this, a lot of disease we are treating, right? So most probably in the future, cancer will also be treated with this way. It is believed that in the coming times, we will have certain plants, we will have certain animals which will replace the medicine, right? Certain plants will be introduced which will replace the medicine. We don't need them to take medicine for sugar. We will just need to take the specific crop, like it will be in, let's suppose in maize, in wheat, it will be inserted, right? The gene will be inserted. So some genes will be turned on and some genes will be turned off. And it has been working. I think for some diseases we have some plants.
Yes.
Right? For some diseases it is still there that there are genetically engineering plants if you are using certain for it. The dicer enzyme then cut this RNA into a microRNA, which is activating splicing complex. So this you take. These complex block the expression of gene producing messenger RNA complementary to microRNA. This has also been fine. RNA interference technology is a powerful way to study gene expression in the laboratory. It also holds the promises of allowing medical scientists to turn off the expression of gene from viruses and cancer cells, and it may provide new way to treat and prevent even cure diseases. If you look here, right, before RNAi technology, before what the scientists were doing, they were eliminating the genes, right? Which genes, like let's suppose a specific gene was going to be studied, right? Let's suppose here in this group you are 20 scientists, right? And you are going to study any organism or any disease, which is bacterial disease, which is viral disease, fungal disease, right? But that fungal disease contains 20 genes. That organism contains 20 genes. So he will study one gene, each and everyone have to study one gene. Let's suppose, right? So what he's going to do, he's going to eliminate 19 genes and study one. Right? Any risk for all in that time. But now what's going to happen? He's going to take simply the RNA for their genes and insert it. So all the other genes will be turned off, but the one gene which he is working, it will remain turned on, right? Or the group of the gene with which he is working for 2, 3, 4, 5, 10, whatever, it will be turned on. So based on these, a lot of disease can be treated, and even the disease which is on the top, that is cancer, right? That if it make it possible, so it will be very major breakthrough. Genetic control of the development. What's control? If you have any question till here, you can ask me. The topic is going to change. Genetic control of development. What controls the development of cell and tissue in the multicellular organism? What do you think? What is going to control the development of cell tissue in a multicellular organism? Genes, right? I told you in the start that before grade 9, you were saying that cell. In grade 9, you also say that cell, right? And in grade 9, you are studying organelles, but I don't know, you study in detail not. Then organelles comes. So we come to cytoplasm. Then inside cytoplasm, we come to nucleus. Then inside nucleus, we come to chromosomes, DNA. And then inside DNA, we come to gene. From gene, we come to microRNA or interference RNA, right? So these are the, you can say, the start of life, which is going to control the things. So overall, we are taking genes, right? Gene is a powerful part of the body, which is going to regulate all the function. It's going to regulate all the expression of the proteins. Master control genes are like switch. They trigger particular patterns of developments and differentiation in a cell and tissue. Here you have a 2-3 example: hog gene, homobox genes, right? These two are given example, but you have to take just general gene, right? Which is responsible for controlling all the factors. Beside this, another point in the science always you have to keep in mind that environmental influence is also going to affect. Environmental influences are important. If you are taking chemistry, physics, and biology. So in these three subjects, what will happen? Environmental influences will be there, right? If it can be in the shape of the air, if it is in the shape of the temperature, right, or anything, but environmental influences are always there. Master control gene are like switch that trigger particular pattern of the development and differentiation in the cell and tissue. Regulating gene expression is especially important in shaping the way a multicellular organism develops, right? So yes, it's the genes expression, right? Like for example, if you take the example of stem cells, right? Stem cell which zygote develop to, right? Zygote give rise to stem cell, right? So the capability of stem cell that it is going to develop into any type, right? So now who is going to decide that this stem cell, let's suppose we have four stem cells, A, B, and C, and D, right? A, B, C, D, right? Cell A is going to convert into skeletal cell, cell B is going to convert into nerve cells, cell C is going to convert into, let's suppose, skin cell, and cell D is going to let's suppose, convert into blood cells, right? So who will decide this? That cell A, B, C are going to develop into different cell gene expression, right? Gene expression we're going to decide it that which type of these stem cell are going to convert. So genes expression, they are going to play a very important role in the development of multicellular organism. Each of the specialized cell types found in the adult organism from the same fertilized egg. As I told you, there's same egg, right? Two chromosomes combined, right? Half from male and half from females. It forms what? It forms zygote, right? Now the cell of the zygote, if it has one cell or if it has 100 cells, they are same, right? So now out of these 50, 60, 100 cells, the entire complex organism is being made. Human, animal, plant, microorganism. From what? From these same cell, same type of cell. It is due to what? It is due to the genes, which is responsible for differentiating the same cell into different type of cells. As an embryo develops, different set of genes are regulated by the transcription factors and repressor. Genes regulating helps cell undergo differentiation, becoming specialized structure and function. The same point as I told you, right? That how they are going to, like from embryo. If you look to the embryo, seed. If you take a seed, right? Seed has embryo, and embryo has all one type of cell, right? Have you seen the seed before? Seed. If you open seed, so inside seed there is an embryo, right? And that embryo is almost the same. It has same type of things. It do not have roots, it do not have stem, it do not have leaves, it do not have fruits, right? Flowers, nothing. But it has only same type of cell. Now they same type of cell are going to be differentiated into different type of cell by their genes. Now we have Aromatic Gene. Edward Lewis was the first to show that a specific group of the genes control the identities of the body parts in the embryo of the common fruit flies, right? So mean that Edward Lewis he studied fruit flies genes, right? The fruit flies genes. It was also discussed by a scientist, a genetist. I think in the time of the Mendel, another scientist also studied fruit fly. No, it was the Mendel and his group, right? Mendel and his group also studied fruit fly. The same experiment they also did. So if you look to that, the first show that the specific group of the genes control the identities of the body parts in the embryo of the common fruit fly. So that was studied by what? By who? That was studied by Edward B. Lewis. Lewis found that the mutation in one of these genes actually related and apply with the legs growing out of its head in the place of the antenna, right? So now the cells, if you look in that of the fruit fly, right? The fruit fly state that the genes which is responsible for the growing of legs, right? So in the fruit flies, right? Instead of antenna, there are what? There are legs. And in the other organism, the same cell are going to develop into what? The same cell are going to develop into antennas. So they are, it depends on what? It depends on the gene expression. That in one organism it is going to develop the same type of cell both have. But in one organism it's going to develop into antenna, and in the other organism it's going to develop into legs. From previous work, it became clear that set of master control genes known as aromatic genes regulate organs that develop in specific part of the body. So from the work of the device, it has been clear that we have a master genes which is known as aromatic genes. It's going to regulate the organ that develop into a specific part of the body. In case of the fruit fly, right? In case of the fruit fly. It is enough, or we need to start the next one. We have box gene and homo box gene. That is a little bit difficult, but it is the last topic.
We have 3 sessions.
But we also have to do the workbook. And one session we will take in chemistry. From biology, we will take one in chemistry. Okay, this is enough for today. Tomorrow we can finish it and we will start the workbook. So bring your workbook for next lesson.
So when you will upload the worksheet?
How much? I prepared almost yesterday. So I will upload it till the end of this week.